course of transcription, the initial RNA product synthesized by RNA Polymerase II enzyme called a primary transcript undergoes several processing steps including capping, splicing and polyadenylation before a functional mRNA, messenger RNA is produced. We shall focus on polyadenylation and splicing in this session. First is polyadenylation. The process of adding polyadenine to 3' end of pre-RNA is called polyadenylation. A long chain of adenosine monophosphate residues is called poly-A. Heterogeneous nuclear mRNA is a precursor to mRNA. Subsequently, it has been shown that the length of actual poly-A tails varies from organism to organism, like in E. coli it is 10 to 40 residues, in yeast more than 90 residues, and in vertebrates approximately 300 residues. In many bacteria, both mRNAs and non-coding RNAs can be polyadenylated. mRNA molecules in both prokaryotes and eukaryotes have polyadenylated 3' ends, with the prokaryotic poly-A tails generally shorter and less mRNA molecules get polyadenylated. Now, polyadenylation in eukaryotes. Eukaryotic cells have 3 types of RNA polymerase, which are called RNA Polymerase I, RNA Polymerase II and RNA Polymerase III. In eukaryotes, each type of RNA is made by its own polymerase, like RNA Polymerase I makes rRNA, RNA Polymerase II makes mRNA and RNA Polymerase III makes tRNA. Moreover, RNAs are synthesized in the nucleus of eukaryotes, but get transported to the cytoplasm for protein synthesis. Nearly all eukaryotic RNA primary transcripts or pre-mRNAs formed by RNA Polymerase II undergo processing before they could actually serve as mRNA for translation or as a component of the translation machinery such as rRNA, 5S RNA, tRNA or RNA processing machinery like snRNAs. It should be noted that the 5S RNA is not processed by capping, polyadenylation or splicing. Next is basic mechanism of polyadenylation. Transcription of eukaryotic genes extends beyond the polyadenylation site. The transcript is cleaved and polyadenylated at the 3' end created by cleavage. Most eukaryotic mRNAs and their precursors have a chain of AMP residues about 250 nucleotides long at their 3' ends. Poly-A tail is added post-transcriptionally by enzyme RNA Polymerase. Now, the cleavage of pre-mRNA. Cleavage in mammals requires several proteins, CPSF, which is cleavage and polyadenylation specificity factor, CstF, which is cleavage stimulation factor, CF1 and CF2, which are cleavage factor 1 and 2, poly-A polymerase and poly-A binding proteins. Now, RNA Polymerase II. The processive polyadenylation complex in the nucleus of eukaryotes work on the product of RNA polymerase II, such as precursor mRNA. The RNA is typically cleaved before transcription termination, as CstF also binds to RNA polymerase II. It signals for RNA polymerase II to slip off the transcript. Now, CpSF, cleavage and polyadenylation specificity factor. It is the first protein to bind to the signaling region near the cleavage site of the pre-mRNA, to which the poly A tail will be added by polynucleotide adenyl transferase. The upstream signaling region has the canonical nucleotide sequence AAUAAA, which is highly conserved across the vast majority of pre-mRNAs. A second downstream signaling region located on the portion of the pre-mRNA that is cleaved before polyadenylation consists of a geo-rich region required for efficient processing. Now, CstF, which is cleavage stimulation factor. Cleavage stimulation factor is 64 kilodalton subunit, a protein that in human is encoded by the CstF2 gene. The protein is a member of the cleavage stimulation factor complex that is involved in the 3' end cleavage and polyadenylation of pre-mRNAs. Specifically, this protein binds geo-rich elements within the 3' untranslated regions of mRNAs. Now, the cleavage factor 1 and 2. Cleavage factor are two closely related proteins involved in the cleavage of 3' signaling region from a newly synthesized pre-messenger RNA molecule in the process of gene transcription. There are two cleavage factors involved in the assembly of protein complex that mediates this process, CF1 and CF2. They are recruited to the cleavage site by cleavage and polyadenylation specificity factor and cleavage stimulatory factor, and form a complex that also includes polyadenine polymerase, which performs the polyadenylation reaction. Poly-A polymerase. Poly-A is synthesized by a nuclear enzyme, Poly-A polymerase. Structures of the enzymes predicted from the longest sequence includes RNA binding domain, polymerase module, 2 nuclear localization signals, and serine threonine rich regions. This is dispensable for in vitro activity. With the help of 2 magnesium ions, it binds to the messenger RNA and adds adenosine nucleotides one at a time to the end of the strand. The structure of Poly-A polymerase has been presented at Protein Data Bank with PTB ID 2Q66, in which the protein is shown in blue surrounding the substrates. A short stretch of Poly-A RNA, 5 nucleotides long, is shown in yellow. ATP is shown in red and a magnesium ion is shown in green. Now, the Poly-A binding protein. Poly-A binding proteins bind to Poly-A tails as soon as they are synthesized in the nucleus, and then they chaperone the messenger RNA throughout its life. The protein is composed of 4 similar domains which bind to a stretch of about 27 RNA nucleotides. Now, the initiation of polyadenylation. 2 proteins participate in the initiation process, Poly-A polymerase and CpSF, which binds to the AAUAAA motif. At first, CpSF binds to the starter sequence, then CstF binds to GU-rich region, and cleavage occurs upstream of CstF binding site. CstF dissociates and short RNAs mimic a newly created mRNA, and 3' end can now be polyadenylated by Poly-A polymerase. When Poly-A reaches about 10 nucleotide in length, further polyadenylation becomes independent of AAUAAA signal and depends on the Poly-A itself. Next is elongation of Poly-A. Elongation of Poly-A in mammals requires a specificity factor called Poly-A binding protein 2, PAB2. This protein binds to a pre-initiated oligo A, aids Poly-A polymerase in the elongation of Poly-A to 250 nucleotides or more. PAB2 acts independently of AAUAA motif. It depends only on Poly-A and its activity is enhanced by CpSF. Not all mRNA molecules are polyadenylated. For example, in animal cells, histone mRNA lacks the Poly-A and the AAUAAA sequence. Next is cytoplasmic polyadenylation. The initial polyadenylation of an RNA molecule occurs in the nucleus apparently automatically for nearly all eukaryotic mRNA precursors. Once in the cytosol, the 200 nucleotide long Poly-A tails on most mRNAs gradually shorten over the course of days. Tails shorter than about 30 A's suggest that this is the minimum tail length required for mRNA stability. Maturing oocytes and eggs provide a striking example of the control of gene expression by means of Poly-A addition to specific mRNAs. Now, the significance of polyadenylation. Poly-A tail aids in the export of mature mRNA from the nucleus through nuclear pores. It seems to serve as a recognition signal for the ribosome that is required for efficient translation of mRNA. Polyadenylation promotes the degradation of a regulatory RNA. Poly-A tracks may lead to mRNA stabilization. With the help of Poly-A binding proteins, it protects the end of RNA strand, shielding it from RNA cutting nucleases. Now, the splicing in eukaryotic RNAs. A sizable fraction of the RNA that is synthesized in the nucleus of eukaryotic cells is not transported to the cytoplasm. This RNA is of a variety of sizes and sequences and is called heterogeneous nuclear RNA. Within this class of RNA are messenger RNA sequences that ultimately are translated into proteins, but they contain non-coding intervening sequences called introns that must be removed by cutting and splicing to form the coding sequence called exons that is found in the mature messenger in the cytoplasm. Rare genes of few viruses, of prokaryotes and of an archaebacterium also contain introns. Now, the biological significance of introns. Introns are highly variable in size, ranging from a few nucleotide pairs to thousands of nucleotide pairs in length. They may play a role in regulating the gene expression. In this way, it is expected to decrease the rate of transcript accumulation in a cell. Introns accumulate new mutations much more rapidly than exons. The discovery by Richard Roberts and Philip Sharps in 1977, Nobel laureate in physiology or medicine in 1993, intervening sequences place genetic spaces between coding regions in a gene. Introns result in multiple products from a single gene. These may be of four types: introns in tRNA genes, group 1 introns, group 2 introns and pre-mRNA introns. Group 1 introns are widespread and present in mRNA, tRNA and rRNA in mitochondria, chloroplast and nuclear genomes of non-vertebrates, in bacteriophages and in eubacterial genomes. Group 2 introns are found in mitochondria and chloroplast, but have also been observed in cyanobacteria and proteobacteria. Next is splicing signal. For genes that encode protein, the splicing mechanism must be precise. It must join exon sequences with accuracy to a single nucleotide to assure that the codons in exons distal to introns are read correctly. This accuracy requires precise splicing signals. Most introns start from the sequence GU, which is referred as splice donor, and end with the sequence AG, which is known as the splice acceptor site in the 5' to 3' direction. Another important sequence is called the branch site, located 20 to 50 bases upstream of the acceptor site. The consensus sequence of the branch site is CU, A or G, A, C or U, where A is conserved in all the genes. In over 60% of the cases, the exon sequence is A or C, AG at the donor site and G at the acceptor site. There are three distinct mechanisms of intron excision from RNA transcripts. The first is splicing of mRNA precursor, pre-mRNA in higher eukaryotes. Unlike group 1 and group 2 introns, pre-mRNA introns are not able to self-splice without the assistance of trans-acting RNA or protein factors. Splicing of nuclear precursor messenger RNA takes place in the spliceosome. These are present in small nuclear RNA protein complexes called snRNPs, small nuclear ribonucleoprotein, pronounced as SNRPs. They contain a set of 5 uridine-rich small RNA molecules called snRNAs, small nuclear RNAs, U1, U2, U4, U5 and U6. These snRNAs do not exist as free RNA molecules. These are transiently associated to more than 70 non-snRNPs, splicing factors, RNA helicases, SR splicing factors, etc. Now, the spliceosome assembly pathway and splicing reaction. Evidence indicates that the U1 snRNP must bind at the 5' splice site prior to the initial cleavage reaction. This binding is primarily the result of base pairing between the 5' splice site consensus sequence and a complementary sequence at the 5' end of U1 snRNA. U2AF recognizes the 3' splice site and binds to the polypyrimidine tract at 3' splice site. It also helps BBP, branch point binding protein, in binding to the branch point site. U2 snRNP added by U2AF, U2AF65 and U2AF35 displaces the BBP and binds to branch site at the consensus sequence that contains the conserved A residue that forms the branch point in the lariat structure of the spliced intron. A pre-assembled tri-snRNP consisting of U4, U6 and U5 snRNP is then incorporated into the complex, bringing the 3 splice sites together to yield the complete spliceosome. In the next step, U1 leaves the complex, replaced by U6 at the 5' splice site. U4 is also released from the complex, allowing U6 to interact with the U2 through RNA-RNA base pairing. The interaction of U2 and U6 creates an active site that juxtaposes the 5' splice site and branch site, facilitating the first trans-esterification reaction. The intron forms the lariat as described earlier. The free exon is held by spliceosome and brought closer to 3' splice site, which is added by U5 snRNP, which align the 2 exons so that they can be joined together. The 2 exon sequences are joined together and the intron along with associated snRNPs is released as a lariat. The excised intron is degraded in the nucleus and snRNPs are recycled. This step requires the hydrolysis of ATP. The spliced mRNA is now ready for export to the cytoplasm and translation on ribosomes. Next is splicing of rRNA precursor pre-rRNA in tetrahymena. It is one of the best examples of autocatalytic splicing or self-splicing, as the introns in rRNA precursors was excised without the involvement of any protein. Moreover, such self-splicing has been shown to occur in rRNA precursors of several lower eukaryotes and in a large number of rRNA, tRNA and mRNA precursors in mitochondria and chloroplast of many different species. Autocatalytic excision of the intron in the tetrahymena rRNA precursor requires no external energy source and no protein. Instead, the splicing involves a series of phosphoester bond transfers with no bonds lost or gained in the process. The reaction requires a guanine nucleoside or nucleotide with a free 3' hydroxyl group, GTP, GDP, GMP or guanosine as a cofactor plus a monovalent cation and a divalent cation. The intron is excised by means of 2 phosphoester bond transfers, and the excised intron can subsequently circularize by means of another phosphoester bond transfer. Next is the splicing of tRNA precursor pre-tRNA in yeast Saccharomyces cerevisiae. The tRNA precursor splicing reaction has been worked out in detail in the yeast Saccharomyces cerevisiae. The introns of tRNA precursors are excised by precise endonucleolytic cleavage and ligation reaction catalyzed by special splicing endonuclease and ligase activities. In stage 1, a nuclear membrane bound splicing endonuclease makes 2 cuts precisely at the end of intron. Cleavage of the tRNA precursor yields 5' hydroxyl termini and 2' 3' cyclic phosphate groups at the 3' termini. The stage 2 ligation process involves 4 separate reactions which are catalyzed by the splicing ligase. Addition of a phosphate group to the 5' OH terminus. This reaction requires kinase activity and a phosphate donor ATP. Then the 5' phosphate group is activated by the transfer of an AMP group to the terminus from an AMP ligase intermediate. The 2' 3' cyclic phosphate is opened by a cyclic phosphodiesterase that produces a 2' phosphate and a free 3' hydroxyl. The final ligation reaction occurs via a nucleophilic attack of the free 3' hydroxyl group on the interior 5' phosphate with the release of AMP. Finally, the 2' phosphate group remaining from the 2' 3' cyclic phosphate produced by the original cleavage reaction is removed by the phosphatase activity to yield the mature tRNA molecule. Now, alternative splicing. In many cases, the splicing process can create a range of unique proteins by varying the exon composition of same mRNA. This phenomena is then called the alternative splicing, which can occur in many ways. Exons can be extended or skipped, or introns can be retained. Alternative splicing generates a tremendous amount of proteomic diversity in humans and significantly affects various functions in cellular processes, tissue specificity, developmental states and diseased conditions. Now, mechanism of alternative splicing. During alternative splicing, cis-acting regulatory elements, enhancers and silencers in the mRNA sequence determine which exons are retained and which exons are spliced out. These cis-acting regulatory elements alter splicing by binding different trans-acting protein factors such as SR, serine-arginine rich proteins that function as splicing facilitators, and HNRNPs heterogeneous nuclear ribonucleoproteins that suppress splicing, the splicing errors and variations. Splicing errors are very rare events which may range from 1 in 1000 or 1 in 1 lakh. Common errors include mutation of a splice site resulting in loss of function of that site, mutation of a splice site reducing specificity, displacement of a splice site leading to inclusion or exclusion of more RNA than expected, resulting in longer or shorter exons. Now, coming to the conclusion, polyadenylation, the addition of poly A tail at the 3' end, and splicing of introns, the non-coding intervening sequences of a gene, are prime processes for the generation of mature, fully functional RNA to get translated into protein.